What if everything you've been told about life's origins was wrong? What if the very fabric of the universe, the precise laws governing reality, the elegant complexity of your own body, even the way your eyes process light, wasn't the result of blind chance, but undeniable evidence of an intelligent designer? Consider this: the human eye, a marvel of engineering more advanced than the most powerful supercomputers, captures millions of pieces of information every second. Its intricate design, from the light-sensitive retina to the brain's rapid-fire processing, raises a staggering question. Could such precision really be the product of random mutation and natural selection? Or does it hint at something far greater, something purposeful? If the human eye, the DNA within every cell, and the cosmic order of the universe all point to a deliberate blueprint, then the implications are profound. Is there a mind behind the universe, an architect shaping the very laws of existence? The answer may challenge everything you believe, and by the time you reach the end of this journey, you might see the world and yourself differently. The marvel of the human eye. A single drop of morning dew clings to the edge of a leaf, reflecting the rising sun in miniature. A hawk, high in the sky, locks its vision on a tiny mouse darting through the grass. A newborn blinks, its wide eyes absorbing the world for the first time. These simple yet profound moments are made possible by one of nature's most intricate and efficient systems, the human eye. When you look into a mirror and examine your own eye, you're not just seeing a reflection of yourself. You're looking at a biological marvel, a living supercomputer designed to interpret the world with stunning accuracy. Beneath the smooth, glass-like cornea lies a complex network that includes over 100 million photoreceptor cells within the retina. These cells convert light into electrical signals, which are sent to the brain via the optic nerve. The human brain, which contains approximately 86 billion neurons, processes this information with remarkable efficiency. The eye doesn't simply see, it decodes, processes, and reconstructs reality in real time, seamlessly adjusting to movement, light, shadow, depth, and color. This constant recalibration happens effortlessly, allowing us to navigate the world with incredible precision. To put the capabilities of the human eye into perspective, consider the most advanced supercomputers of today. Leading systems such as IBM's Summit, Google's Sycamore, and NVIDIA's DGX supercomputers are capable of processing vast amounts of data at impressive speeds. However, even these powerful machines are outclassed by the processing efficiency of the human retina. Research has shown that the human retina, which contains approximately 100 million photoreceptors, can process visual information in real time with extraordinary efficiency. In fact, it has been estimated that the human eye can process an enormous amount of visual data each second, more than a supercomputer can handle in the same amount of time. For instance, a supercomputer operating at petaflop speeds, 1 quadrillion calculations per second, would require considerable time to replicate even a second of the retina's real-time processing of light, depth, color, and movement. Despite the advanced computational power of AI systems and supercomputers, these technologies still fall short when compared to the retina's ability to process dynamic visual information. Artificial intelligence, even on GPUs designed for deep learning, is still not able to match the retina's seamless processing of visual data as it adjusts to changes in the environment. Given that supercomputers, the product of intelligent design, still fall short of replicating the retina's performance, we must ask, How did such a sophisticated system come to be? Did this incredibly advanced visual processing system emerge randomly through mutations and natural selection, or does it suggest the work of a masterful designer? This question invites deep reflection and exploration. The eye is an astonishing marvel, but its brilliance would be meaningless without the microscopic world that powers it. Every blink, every shift in focus, every electrical signal that races through the optic nerve depends on something even more fundamental: the molecules that make life possible. Deep within every living cell, far smaller than even the most advanced microscope can see, lies a language. A code written not in ink or electricity, but in chemical sequences. This is the language of proteins, the essential workers of life. They build our muscles, fight disease, transport oxygen, and enable the very act of sight. And yet, these tiny molecular machines are so intricately structured that their existence raises a question few dare to ask. Can pure chance really explain the precise organization of life's building blocks? Proteins are not simple molecules. They are informational macromolecules, built from 20 different amino acids arranged in exact, purposeful sequences. These sequences are not random. They must be ordered in just the right way for each protein to function correctly. The average protein consists of a chain of about 500 amino acids, and here's where the numbers become staggering. If you were to randomly assemble a protein by stringing together 500 amino acids, the number of possible combinations would be beyond human comprehension, one followed by 600 zeros. That is a number so vast that it exceeds the total number of atoms in the known universe. Even if nature had billions of years to experiment, could it really stumble upon the correct sequences? Not just once, but for thousands of different essential proteins without any guiding intelligence. To put this challenge into perspective, imagine trying to form a simple English phrase by random chance. Let's take a short sentence. The theory of evolution. This phrase has 23 letters and spaces, and if we were to attempt to form it by randomly pulling characters from a Scrabble set containing 26 letters plus a space, the probability of getting the correct phrase would be 1 in 800 million trillion tries. Now suppose we speed up the process and pull characters at the rate of 1 billion per second. Even at this unimaginable speed, it would take about 26,000 trillion years to produce the correct sequence just once. And yet this phrase is trivial compared to the complexity of even the smallest functional protein. If assembling a simple sentence at random is practically impossible, how can we believe that life's far more intricate molecular sequences arose by sheer happenstance? Every protein in your body is a carefully ordered structure, precisely arranged to perform its task with mind-boggling efficiency. The instructions for assembling these proteins are embedded in your DNA, a code so advanced that it far surpasses any human-made programming language. So how did this code come into being? Did nature blindly stumble upon billions of precise functional sequences against impossible odds? Or does the very existence of this biological language suggest an intelligent author, one who wrote the blueprint of life with purpose and intent? Imagine standing on the edge of a vast canyon at midnight, looking up at an endless sea of stars. The silence is overwhelming, the vastness humbling. Billions of galaxies stretch across the cosmos, each one a swirling masterpiece of light and motion. But what if I told you that all of it, every planet, every star, every law of physics, exists on the razor's edge of impossibility? The deeper we look into the universe, the clearer it becomes. Everything is balanced with such incredible precision that even the slightest shift would unravel existence itself. For life to exist, the universe must obey a very specific set of physical laws, constants, and forces. These fundamental numbers, the gravitational constant, the strength of the electromagnetic force, the cosmological constant, govern everything from the structure of atoms to the movement of galaxies. But here's the astonishing part. These numbers are so precisely tuned that even the tiniest variation would render the universe uninhabitable. If gravity were just slightly stronger, stars would burn out too quickly, collapsing into black holes before life had a chance to form. If gravity were just slightly weaker, galaxies would never hold together, and planets, along with the conditions necessary for life, would never form. If the electromagnetic force were altered even slightly, the chemistry of life would become impossible. Atoms would not bond properly, and molecules, including DNA, could not exist. If the cosmological constant, which controls the expansion of the universe, were even a fraction different, the universe would have either collapsed in on itself or expanded too quickly for galaxies to form. The margin for error is so unfathomably small that it defies coincidence. Imagine dialing a combination lock with trillions of individual dials, each one needing to be set to the exact right position for the lock to open. That is the level of precision we are dealing with. Some scientists have tried to explain this fine-tuning by suggesting we just got lucky, that out of countless possible universes, we just happen to exist in the one where all the numbers are right. But luck cannot explain precision at this level. The odds are so astronomically small that chance is not a rational answer. Consider this. If you walked into an abandoned cabin in the woods and found a table perfectly set for dinner with a warm meal waiting, would you assume the wind randomly placed the plates and lit the candles, or would you conclude that someone had prepared it with intention? The same reasoning applies to the cosmos. The fine-tuning of the universe looks far less like an accident and far more like the work of a masterful designer. The question we must ask is this: if the laws of physics themselves appear to be calibrated for life, could it be that the universe was intended for us to be here? The deeper we go, the more the universe begins to look less like a cosmic accident and more like a carefully constructed home, one built with purpose, precision, and intent. The universe is fine-tuned for life, but life itself is even more astonishing. While the cosmos follows precise mathematical laws, within every living cell lies something just as breathtaking: a code, a blueprint, a language. And not just any language, but one so advanced, so intricately arranged that it rivals the most sophisticated computer programs ever written. This is the code of DNA. If you were to shrink down into the microscopic world of a single human cell, you would find something extraordinary. Twisting inside the nucleus of every one of your cells is DNA, a molecule that carries the complete set of instructions for building and maintaining your body. But DNA is not just a chemical structure, it is a digital, information-rich system, precisely ordered and highly complex. DNA is composed of 4 nucleotide bases: adenine, thymine, cytosine, and guanine, which are arranged in an exact sequence, much like the ones and zeros in a computer program. This sequence functions as a biological software code, spelling out instructions that determine everything from your eye color to how your body heals itself. A single human cell contains about 3 billion letters of this genetic code, enough information to fill 1,000 encyclopedias. And yet, every letter, every sequence, every bit of information must be precisely ordered for the cell to function properly. Let's put this in perspective. Imagine stumbling upon a book written in perfect English, its sentences clear, its paragraphs meaningful, its structure flawless. Would you assume that the ink randomly splattered onto the pages over millions of years, forming words by pure chance? Or would you recognize that someone, an intelligent mind, deliberately wrote it? Even the simplest bacteria contain genetic instructions more complex than any software program humans have ever written. If we found an alien probe on Mars covered in meaningful symbols, we would immediately assume it was designed by an intelligent being. Why then do we assume that DNA, far more complex and functional, arose by accident? In engineered systems, digital information does not arise randomly. It is the result of intentional design. Every computer program, every coded message, every structured language in history has been written by a mind. If DNA is also a highly structured, information-rich code, then the most logical question we can ask is, who wrote it? Scientists who reject intelligent design often claim that the genetic code somehow evolved over time through random mutation and natural selection. But no known process, natural or artificial, has ever demonstrated the ability to generate functional, meaningful digital information without input from an intelligent source. If we found a message written in perfect code, we would never assume it came from random forces. And yet, inside every living cell, we find something even more astonishing, a code far beyond anything humanity has ever created. DNA carries the blueprint of life, a structured, functional, and information-driven system embedded in every cell of every living organism. The deeper we examine life, the more we encounter structures so intricately engineered that they defy traditional evolutionary explanations. Some systems within living organisms are so interwoven, so reliant on each component working in perfect harmony, that if even one part were removed, the entire function would collapse. This concept is known as irreducible complexity, and it presents one of the most profound challenges to the idea that life developed solely through small, incremental changes. Consider one of the simplest organisms on Earth, bacteria. At first glance, bacteria seem primitive, nothing more than single-celled life forms drifting in their microscopic world. But some bacteria possess a flagellum, a whip-like tail that rotates like an outboard motor, propelling them forward. This flagellum is not just a random appendage, it is a highly complex machine. It consists of dozens of precisely arranged proteins, including a rotor, a stator, bushings, and even a universal joint. The flagellum can rotate at speeds of up to 100,000 revolutions per minute, reversing direction in a fraction of a second. It operates much like an electric motor, complete with self-assembling parts and an energy-efficient design so advanced that engineers study it for inspiration. Here's the problem for traditional evolutionary theory. If even one of the essential protein components of the flagellum is missing, the entire system fails. There is no stepwise, gradual pathway that allows for half a flagellum to be functional. It must be fully assembled, with every part in place, to work at all. How could such an intricate system arise piece by piece, when the individual pieces are useless without the whole? This is the very definition of irreducible complexity, a feature that suggests a system designed as a complete unit rather than assembled randomly over time. Now, consider something much closer to home, your own bloodstream. Every time you get a small cut, your body springs into action, activating a sophisticated blood-clotting cascade. This process is not as simple as sealing a leak with a patch. Rather, it involves a delicate chain reaction of specialized proteins, each triggering the next in a precise sequence. If the sequence were missing just one step, the process would fail. If clotting happened too soon, your blood would thicken and cause deadly blockages. If it happened too late, even the smallest wound could result in fatal bleeding. The balance is perfect, finely tuned to keep you alive. Could such a system have evolved gradually with incomplete steps that had no function along the way? Or does it make more sense that this life-preserving mechanism was put in place all at once intentionally? Every cell in your body is a bustling metropolis, filled with machines assembling the materials of life. At the heart of this microscopic world is the ribosome, an astonishingly complex molecular factory. The ribosome's job is to read the genetic instructions in your DNA and build proteins, the very building blocks of life itself. It operates with extreme precision, ensuring that amino acids are arranged in exactly the right order to form functional proteins. Here's the puzzle. The ribosome is made of proteins, but it is also needed to make proteins. This creates a classic chicken or the egg problem. How could the first ribosome exist if it needed proteins to build itself? No working ribosome, no proteins, no ribosome. Evolutionary explanations struggle to account for this circular dependency. But it aligns perfectly with the idea of an intelligent designer, setting everything in motion at once with all necessary parts in place. If these biological systems were found in a machine or a factory, no one would assume they were the result of chance. Engineers know that functional complexity does not arise randomly. It requires planning, foresight, and precise coordination. So why do we assume that nature, which far surpasses human technology in complexity, came into existence without a designer? Irreducible complexity presents a profound challenge. If life is filled with interdependent, purpose-driven systems that cannot function unless fully formed, could they really have emerged by slow, unguided processes? Or does life, in all its breathtaking intricacy, point unmistakably to the work of an intelligent architect? The Cambrian explosion, a burst of design. Scientists believe that for millions of years, life on Earth remained simple. The fossil record reveals only single-celled organisms and a few soft-bodied creatures drifting through ancient oceans. Then something extraordinary happened, something so sudden, so dramatic, that it defies conventional evolutionary explanations. In a geological instant, the seas erupted with life. Entirely new creatures, each with fully formed functional body plans, appeared in the fossil record without clear evolutionary ancestors. This event, known as the Cambrian explosion, remains one of the greatest mysteries in the history of life. Over 500 million years ago, life underwent an unprecedented transformation. Fossil beds from this era reveal a stunning diversity of organisms, including some of the earliest appearances of nearly every major animal group we recognize today. Trilobites, complex armored arthropods with advanced eyes and jointed limbs, Anomalocaris, a massive predator with rotating mouthparts and highly sensitive appendages, Opabinia, a bizarre five-eyed creature with a flexible grasping trunk, Waiwaxia, a spiky, plated invertebrate that baffled scientists with its intricate structure. Before this, nothing like these creatures existed. The leap from simple single-celled life to fully developed animals with muscles, nervous systems, exoskeletons, and eyes seemed nearly instantaneous in evolutionary terms. Where were the small, gradual changes that should have built these species step by step? Where were the transitional forms? Even Charles Darwin was troubled by the Cambrian explosion. His theory of gradual evolution predicted that life should develop slowly, with countless intermediate forms bridging the gaps between major groups. But the fossil record told a different story. Instead of a long chain of small modifications, scientists found fully formed, highly complex organisms appearing abruptly, as if they had been introduced all at once. Darwin hoped future fossil discoveries would fill in the missing links, but over 150 years later, the gaps remain. If anything, new evidence has made the Cambrian explosion even more puzzling. Complexity arrived too fast for standard evolutionary explanations. Many species appear without clear ancestors, breaking the expected tree of life. The genetic information needed to build these creatures must have existed before their sudden appearance. Where did the genetic instructions for these new body plans come from? Did natural selection somehow preload massive amounts of biological information in anticipation of this sudden diversification? Or is there a more compelling explanation? Some researchers suggest that the Cambrian explosion was not the result of blind evolutionary forces alone, but rather the unfolding of pre-designed biological blueprints, information embedded in life from the very beginning. Consider how technology advances. We don't develop new machines by blindly assembling parts until something works. Instead, engineers design reusable components, motors, sensors, processors that can be adapted for different functions. Could life itself have followed a similar pattern? What if instead of a purely random, stepwise evolution, the rapid appearance of these organisms suggests a modular, pre-existing set of biological components? The genetic coding for advanced body structures may have already been present, waiting for the right conditions to activate. The sudden diversification of Cambrian species resembles an engineered expansion rather than an accidental mutation-driven process. The introduction of fully functional, interdependent systems suggests design much like an architect implementing a master blueprint. This idea would explain why new phyla, completely distinct body plans, appear all at once, rather than emerging gradually over eons. It would also align with what we know about biological information. Functional complexity does not emerge from randomness. It is assembled with purpose. The Cambrian explosion remains one of the greatest mysteries in science. But one thing is certain, it does not fit neatly into the standard narrative of gradual evolution. The sudden, fully functional emergence of life's complexity suggests that something more may be at play, something intentional. If every machine requires a designer and every blueprint requires an architect, then what does the Cambrian explosion tell us about the origins of life? Perhaps life did not merely stumble into existence. Perhaps it was unveiled, bursting onto the scene like a masterpiece waiting for its moment. Inside a sterile, brightly lit laboratory, a team of scientists huddle over a complex array of beakers, test tubes, and high-tech equipment. The air is thick with anticipation, the faint hum of machines filling the space as they attempt to recreate the greatest mystery of all, the origin of life itself. This is not science fiction. For decades, researchers around the world have been striving to answer a fundamental question. Can life emerge from purely natural, unguided processes? Can the raw ingredients of the universe, carbon, nitrogen, oxygen, somehow organize themselves into a self-replicating, information-driven system like a living cell? The stakes could not be higher. If scientists can demonstrate a clear, step-by-step path from simple chemicals to the first functional, self-replicating organism, it would provide the ultimate confirmation that life arose without design. But if they cannot, then the very foundations of materialistic explanations for life's origins begin to crumble. The concept of abiogenesis, The idea that life arose from non-living matter has long been an area of intense research. Many experiments have been conducted to simulate the conditions of the early Earth, seeking to generate the building blocks of life. In 1953, the famous Miller-Urey experiment attempted to replicate Earth's primordial conditions by passing electrical sparks, simulating lightning, through a mixture of gases thought to represent the early atmosphere. The result? A few simple amino acids, the fundamental components of proteins, were formed. Since then, researchers have repeated and refined these experiments, producing various organic molecules like sugars, lipids, and even nucleotides, the subunits of DNA and RNA. But despite these promising beginnings, scientists have never come close to producing a self-replicating system, let alone something as sophisticated as a living cell. And this is where the real problem begins. Creating the building blocks of life is one thing. Assembling them into a working, self-sustaining system is an entirely different challenge. A cell is not just a collection of molecules floating in a primordial soup. It is a highly organized, information-driven structure. Every living cell contains DNA, a storage system encoding precise instructions for building proteins, ribosomes, complex molecular machines that translate DNA's instructions and assemble proteins with pinpoint accuracy, membranes, sophisticated barriers that regulate what enters and exits, maintaining the delicate balance required for survival. Metabolic pathways, intricate chemical circuits that convert energy into usable forms, keeping the cell alive and functioning. For abiogenesis to be true, all of these interdependent systems must emerge together, in the right place, at the right time, functioning in perfect harmony. The challenge is not just forming the pieces, but arranging them into a fully operational system. Here's where naturalistic explanations face a staggering dilemma. If DNA comes first, what reads it? Without ribosomes, it is just a meaningless sequence of chemicals. If proteins come first, what builds them? Without DNA, there is no instruction set for their creation. If cell membranes form first, what do they protect? Without genetic information and metabolic functions, they enclose nothing but lifeless molecules. The emergence of a functional cell is an all-or-nothing event. It is like trying to assemble a complex machine without instructions, without tools, and without a guiding hand, and yet somehow expecting the machine to build itself. Despite decades of research, the field of abiogenesis has yet to demonstrate a clear, viable pathway from non-life to life. Some scientists suggest that billions of years of random interactions might have eventually led to self-replicating molecules, but even this explanation faces massive hurdles. In reality, even the simplest living cell is far more complex than any human-made factory, software program, or mechanical system. And every known instance of complex, functional systems requires intelligence to bring them into existence. If engineers cannot build a self-replicating machine without a blueprint, why should we believe nature somehow did it on its own? If life's origin was simple, scientists should have replicated it by now. Instead, research reveals increasing complexity, defying materialistic explanations. Was life deliberately set in motion? Does its intricate design suggest intelligence? For now, the search continues, but the mystery deepens, resisting chance as an answer. Imagine walking into an ancient library, its towering shelves filled with volumes of knowledge written over millennia. Every book contains meticulously arranged information, stories, blueprints, instructions, each one built upon the discoveries of the past. This library does not grow by accident. New knowledge is carefully added, organized, and preserved by those who understand its value. Now consider this. Life itself operates like this expanding library, continuously accumulating functional information over time. But where does this information come from? How is it stored, transmitted, and refined? And most importantly, can purely random, unguided processes explain its increasing complexity? From the moment of conception, an organism's DNA carries the instructions for its development, growth, and survival. This information is not just present, it accumulates across generations. New biological features, more refined cellular processes, and increasingly sophisticated molecular systems all emerge over time. But here's the critical challenge. Functional information does not just exist, it must be generated and maintained in a meaningful, structured way. In a computer system, functional information is added through intelligent programming. In written language, meaningful content grows through careful authorship. In biological systems, new structures and functions emerge, but can this really happen without an external guiding intelligence? Every known example of an information-based system, whether digital, linguistic, or mechanical, requires an intelligent source to introduce and refine new functional data. So why would we assume life's information is different? Traditional evolutionary theory argues that random mutations, small accidental changes in DNA, can lead to the steady accumulation of new beneficial traits. Over time, these changes are said to shape organisms into more complex and well-adapted forms. But random mutations, by their very nature, are overwhelmingly destructive or neutral, not creative. Destructive mutations introduce errors that cause disease or dysfunction. Neutral mutations have no impact and accumulate as junk in the genome. Beneficial mutations, when they do occur, are often minor and rarely introduce entirely new structures or functions. If evolution were truly driven by random mutations, we would expect to see increasing genetic disorder over time, not the rise of more sophisticated, highly functional systems. The mere existence of new features is not enough. There must be a mechanism that organizes and directs this accumulation of functional. Imagine walking into a vast library. Instead of books, the shelves are filled with meticulously organized volumes of life. Each volume represents a different species, and within each volume lies a detailed blueprint for its construction and operation. This library is constantly expanding, with new volumes appearing over time, representing the evolution of life on Earth. This is the library of life, and its existence begs the question, how did such an intricate and complex collection come to be? This library didn't appear overnight. It has been built over billions of years, with each new generation adding its own chapter to the ever-growing narrative of life. But unlike a library curated by human hands, the library of life is said to have been shaped by the blind forces of natural selection acting on random mutations. This raises a fundamental question. Can a purely unguided process create the intricate order and complexity we see in living organisms? Just as a library requires librarians and authors to organize and write its books, could life on Earth require some form of guiding intelligence to assemble its intricate designs? This essay will explore the fascinating world of biology through the lens of information, examining whether the unguided mechanism of evolution can fully account for the library of life, or if a more profound explanation is needed. The sheer diversity and complexity of life housed within this metaphorical library point towards a process that goes beyond simple chance. Like a well-written novel, life exhibits a depth and intricacy that suggests an author, a designer, a guiding hand behind its creation. At the heart of every living cell lies an elegant molecule called DNA. This molecule serves as the blueprint of life, containing the instructions needed to build and maintain an organism. These instructions are encoded within DNA's unique structure, a double helix resembling a twisted ladder. The rungs of this ladder are formed by pairs of chemical bases: adenine, thymine, guanine, and cytosine. The precise sequence of these bases forms the genetic code, much like the arrangement of letters forms words and sentences. Just as a sentence conveys meaning, the sequence of bases in DNA carries instructions for building proteins, the workhorses of the cell. These proteins carry out a vast array of functions, from catalyzing metabolic reactions to transporting molecules and providing structural support. From the moment of conception, DNA's instructions are faithfully copied and passed down from one generation to the next. This process ensures the continuity of life, allowing traits to be inherited and species to persist over time. DNA's ability to store and transmit information is essential for life's existence, acting as a biological hard drive that preserves the accumulated wisdom of billions of years of evolution. However, DNA is more than just a passive storage device. It's a dynamic blueprint that can be modified over time through mutations, changes in the DNA sequence. These mutations can be caused by various factors, such as errors during DNA replication or exposure to environmental mutagens. While some mutations can be harmful or even fatal, others can be beneficial, providing the raw material for natural selection to act upon. Proponents of unguided evolution argue that random mutations, coupled with natural selection, are sufficient to explain the complexity of life. According to this view, mutations provide the variation, and natural selection acts as a filter, preserving beneficial changes and eliminating detrimental ones. Over vast stretches of time, this process is said to have gradually sculpted simple life forms into the intricate organisms we see today. However, the sheer improbability of random mutations generating complex features raises significant questions about the adequacy of this explanation. Consider the human eye, a marvel of biological engineering, with its intricate lens, iris, retina, and optic nerve, all working in harmony to provide us with sight. To suggest that such an intricate system arose through a series of random mutations, each one conferring a slight advantage and gradually building upon the previous one, stretches the limits of credulity. The odds of such a process producing a structure as complex as the eye are astronomically small, akin to a hurricane assembling a Boeing 747 by chance from a junkyard of airplane parts. The sheer number of mutations required to transform a light-sensitive patch of cells into a fully functional eye, along with the intricate coordination between different components, suggests that something more than random chance is at play. This problem is not limited to the eye. Throughout nature, we find countless examples of irreducible complexity, where biological systems are composed of multiple interacting parts that are all essential for the system to function. Remove any one part and the entire system ceases to work. This intricate interdependence of parts poses a significant challenge to the notion of gradual evolution through random mutations. One of the most remarkable features of DNA is its ability to conserve and transmit functional information over generations. Just as a library protects its valuable books from damage and decay, living organisms possess intricate mechanisms to safeguard the integrity of their genetic information. These mechanisms include DNA repair systems that constantly scan for and correct errors in the DNA sequence, ensuring that the instructions for life remain intact. Furthermore, the process of DNA replication, while remarkably accurate, is not perfect. Occasionally, errors slip through, introducing mutations into the genetic code. While some of these mutations may be harmful, others can be neutral or even beneficial, providing the raw material for natural selection to act upon. This constant interplay between mutation and selection drives the process of evolution, allowing populations to adapt to changing environments over time. However, the key point here is that functional information, once it arises, tends to be conserved. Just as a valuable book in a library is treasured and preserved, so too are beneficial mutations in the DNA sequence. Natural selection acts as a curator, weeding out harmful mutations while preserving those that enhance an organism's chances of survival and reproduction. This conservation of functional information is a testament to the power of natural selection to preserve and build upon existing genetic information. However, it does not address the fundamental question of how functional information arises in the first place. Can random mutations truly generate the novel genes and proteins that underlie new biological features, or is a more intelligent process required? To understand the challenges of explaining biological complexity through unguided processes, it's helpful to draw an analogy from the world of computer science. Just as DNA contains the instructions for building and operating a living organism, software code provides the instructions for running a computer program. Both systems rely on information encoded in a specific language to carry out their functions. Imagine finding a complex software program such as an operating system or a video game, running flawlessly on a computer. You examine the code, and marvel at its elegance and efficiency. Would you conclude that this sophisticated program arose spontaneously through a series of random changes to the code, or would you infer the involvement of an intelligent programmer? The same logic applies to the biological world. The intricate machinery of the cell with its complex metabolic pathways, protein synthesis machinery, and DNA replication system rivals the complexity of any human-designed software. To suggest that such a system could arise through random mutations and natural selection alone stretches the limits of credibility. The concept of specified complexity, often used in discussions of intelligent design, argues that certain patterns or structures exhibit a high degree of specificity and complexity that cannot be explained by chance alone. Just as a random sequence of letters is unlikely to produce a meaningful sentence, a random arrangement of amino acids is highly improbable to result in a functional protein. The evidence presented thus far points towards a profound question. Is the intelligence we observe in the design of software and other human artifacts a reflection of a deeper intelligence embedded within the fabric of life itself? Could it be that the intricate information encoded in DNA, the blueprint of life, is not the product of blind chance, but rather the result of a guiding hand, an intelligence that transcends the material realm? This is where the concept of intelligent design enters the discussion. Intelligent design is not a religious-based argument, but rather an inference from the evidence of nature itself. It posits that certain features of the natural world are best explained by an intelligent cause, not an undirected process like natural selection acting on random variations. The argument for intelligent design rests on the observation that intelligent agents consistently produce large amounts of specified information. We see this in our own lives, from the books we read to the buildings we inhabit. Information in this sense is not merely complexity, but rather complexity arranged in a specific way that conveys meaning and function. Proponents of intelligent design argue that the same principle applies to the biological world. The vast amounts of specified information encoded in DNA, the intricate molecular machines within cells, and the irreducible complexity of many biological systems all point towards the involvement of an intelligent agent in the origin and development of life. As we stand in awe of the library of life, marveling at its vastness and intricate organization, we are left with more questions than answers. Can the unguided mechanisms of evolution truly account for the origin and diversity of life? Or does the evidence point towards a more profound explanation? The Cambrian explosion, a period of rapid diversification of life forms that occurred approximately 540 million years ago, remains a significant challenge to traditional evolutionary theory. The sudden appearance of numerous complex body plans in a relatively short geological time frame suggests that the mechanisms of mutation and natural selection may not be sufficient to explain such rapid evolutionary innovation. Furthermore, despite decades of research, attempts to recreate the conditions necessary for life to arise from non-living matter in laboratory settings have consistently fallen short. The failure of abiogenesis experiments to produce even the simplest self-replicating molecules casts further doubt on the adequacy of purely materialistic explanations for the origin of life. While the debate over the origin and development of life continues, one thing is certain: the library of life is a testament to the power of information. Whether this information is the product of blind chance or intelligent design is a question that continues to captivate scientists, philosophers, and theologians alike. As we delve deeper into the mysteries of life, we must remain open to the possibility that the answer may lie beyond the realm of what we can currently observe or fully comprehend.
Thanks for watching. If you have questions about a Bible topic, feel free to ask in the comments. Don't forget to subscribe and check out these other videos.